ladies and gentlemen. Welcome to English for Medical Postgraduates Reading and Writing course. Today, my lecture will focus on ethical issues in the use of animals in biomedical research. So let's start. After a lead-in, we will explore the theme of this essay, followed by intensive study of this text and critical reflection about the topic. So, are you familiar with this picture? Of course, as medical students, you must know very clearly about this little mouse. He has an ear grown on the back of the mouse. So, animals are widely used in biomedical research, and I would like you to share your experience of animal experimentation. So, have you ever tested on animals? And what were their reactions? I bet most of you must still remember the first struggle and the first scream of the animal in your first experiment. Right. If animals can be experimented on, we can see that this little mouse here might not mind having an ear on the back because he can have better sentiments about sounds. But what about this little mouse here? He has some pieces of human tumors transplanted into her body. And this one here, he is sedated and scanned with an ultrasound to see how well his cancer is responding to treatment. Well, here comes naturally this question. Do animals have the ability of feeling pain or even pleasure? And if they do they have the psychological experiences of fear and irritation? And if the answer to that question is also positive, here we come another question. That is, do they share fraternity with human beings? And if they do, here comes the ultimate question. Why are we justified? Or how can we provide moral justification for animal experimentation? So, let's come to an introduction about this topic, moral justifications of animal experimentation. Actually, I have invited a professor from Cambridge University to explain to us about the benefits brought about by animal experimentation. So, I'm going to play you the video and invite you to answer 3 questions. First one is, what are the mice in Cambridge labs used for? Secondly, what is the only way of understanding how tissues go wrong? And thirdly, why was mice chosen as experimental animals?
Animal research plays a vital role in furthering our understanding of how our bodies work and what causes diseases. Here, at one of the University of Cambridge's animal facilities, there are around 5,000 mice. These mice are helping our researchers in the fight against cancer, a disease that will affect half of us at some point in our lives.
I'm Gerald Evan. I'm a professor of biochemistry, and I'm interested in the molecular basis of cancer, and I've spent all my professional life trying to understand what makes cancers tick. In particular, my laboratory is interested in 2 rather unpleasant cancers. All cancers are unpleasant, but these are particularly nasty ones. Cancers of the pancreas and cancer of the lung. So cancers are diseases where tissues go wrong, not just cells. So in order to understand how tissues go wrong, we have to have all the cells that are in that deranged tissue and understand how they're all talking to one another and interacting with each other. And the only way that we know of doing that is actually in living tissues as they are. We can't recapitulate that in bottles. Mice are the closest that we can get experimentally to human beings. Obviously we could get much closer, but then there will be even more problems associated with working with organisms that, if you like, are more intelligent, closer to human beings. And also mice are relatively easy to breed and maintain and keep going. So they're the best experimental system that we have, even though we would of course prefer not to use them.
Okay, you must have found the answers to the 3 questions, right? So the first question, mice is used in Cambridge labs to fight against cancer, right? That is a horrible word, right? Humans hate that. And the second question, the only way of understanding how tissues go wrong is to observe these tissues in living organisms instead of in bottles. But the question comes, we cannot experiment on human beings, so we have to resort to animals. And the third question, why is mice chosen? Actually, the professor provides 2 explanations. Firstly, they are the closest we can get experimentally to human beings. And secondly, they are small and easy to breed and maintain. Yes. So it is obvious for us that there are enormous benefits brought by animal experimentation. So that's why animal experiments can be used to develop new medicines and also to test the safety of other consumer products. And people supporting animal experimentation always emphasize that animals can only be experimented on if and only if suffering is minimized and human benefits are gained. However, there are also people opposing animal experimentation for the following reasons. They think that animal experimentation is always unacceptable because, firstly, it causes suffering to animals inevitably, and secondly, the benefits to human beings are not always valid. But we're sacrificing the innocent animals. And lastly, any benefits brought by animal experimentation actually could be conducted in other ways, for example, computer modeling and also cell culture. So it seems that the debate will still continue, but I will call your attention to a stop. Let's pause about the debate and come back to look at the principles we abide by while we are experimenting on animals. Currently, we advocate the 3R principle. They are the principles that scientists are encouraged to follow in order to reduce the impact of research on animals. They are reduction, that is, reducing the number of animals used in experiments. Refinement, that is, refining the experiment or the way the animals are cared. And lastly, replacement, that is, replacing experiments on animals with alternative techniques, as we mentioned just now, like cell culture and computer techniques. So now how about we stop arguing about whether animals should be used in experiments? How about we stop and pause and take a historical overview of the arguments and debates that are involved in experimenting on animals? It seems that the debate will not settle. So why don't we take a look back at the journey we have gone so far? Actually, the use of animals in biomedical research can be traced back to more than 2,000 years ago, since the birth of medicine.